Thanks so much for the introduction, and again, thank you all for joining. We're really happy to talk to everyone today. We've had a lot of interesting work that's come through the company this past year. Specifically, after we start a short introduction of our technology, we're going to highlight a number of our recent papers and projects, from antibody validation to the translation of sequencing data into personalized neoepitope microarrays for anti-tumor antibody profiling. So specifically, in more detail, some of the applications we're going to cover is with PEPperChip peptide microarrays. We can monitor the humoral immune response to selected tumor antigens, identify new cancer epitopes from antigens or DNA mutations, determine cancer-stimulating autoantibodies, investigate the influence of tumor treatment on antibody responses, validate therapeutic antibodies for cancer treatment, and discover prognostic tumor markers. But why should you work with our laser-printed peptide microarrays? Well, in contrast to other providers which work with standard spotting technologies, there's no need to pre-synthesize every peptide for the microarray. This means we can save a remarkable amount of the cost for peptide microarray synthesis. Moreover, we can print significantly more peptides per chip for high-resolution epitope mapping, and we're completely flexible with respect to the peptide content of the microarray. So how does the technology work? Well, instead of liquids, we're working with small toner particles that contain amino acids for combinatorial peptide synthesis. So the amino acid toners are printed with custom peptide laser directly onto glass slides. So this slide is a little bit busy, but up here in the upper right corner, this is actually a picture of our printer that we have in our headquarters in Germany. It's about 4 to 5 times the size of a normal office laser printer. And here on the lower right-hand corner is a picture of the cartridges. So instead of having 4 color cartridges, we have 24, each for the one for each of the natural amino acids and 4 for unnatural amino acids. So how it works is there's a conveyor belt that goes through this machine, and we start by printing the, after melting, the residual toner material is washed away, and the rest is standard solid phase peptide synthesis with deprotection of the N-terminal protection group and printing of the next laser amino acid toners. That way we can print up to 1,000 different peptides on centimeters squared with very low material consumption. And what's important, cost factor compared to spotting technologies. Due to the digital printing flexibility of the peptide laser printer, it doesn't make a difference for us if we print 100 times the very same or 100 different peptide microarrays. And then last but not least, we provide a very high peptide quality due to routine double coupling steps and fast production times. So in the upper right corner, this is a schematic of how it works. You have your glass slide, you print the amino acid, you melt, couple, you wash, deprotect, and then keep going over and over until you have your linear peptide printed onto the glass slide. So besides the standard format, middle left, which is right here, and this is about the size, or it is the size of a normal microscope slide that you would see in the lab. And it covers up to 11,000 peptide features. But we also cover a discovery format on the left with up to 70,000 features per microarray, as well as multiplex formats with up to 16 array copies per microarray and 240 peptides features each array copy. So we provide either peptide microarrays including all microarray accessories for assays in your labs, or comprehensive service solutions from epitope mapping to epitope analysis to antigen, epitope, and peptide discovery. So as mentioned kind of briefly, since we have the direct spotting onto the glass slides, we outperform in speed and also our flexibility, quality, reproducibility. We also do conformational or cyclic constrained peptides, which I'll talk a little bit more in detail later. And economics, we pay per chip, not per peptide. So our laser-printed peptide microarrays can be applied in various research areas ranging from infectious disease to autoimmune research to vaccine and peptide drug development. So for the validation of therapeutic antibodies in cancer immunotherapy, we also provide a number of solutions. So our microarrays can be used for linear or conformational epitope mapping with the highest amino acid resolution from single to multiple antigens, or more or less all kind of samples from purified antibodies to hybridomas supernatants, human sera, or plasma samples. Once the epitope is known, we offer epitope substitution scan to narrow down the essential and variable amino acids of an epitope and to perform a bioinformatic cross-reactivity analysis of a diagnostic or therapeutic antibody. So this option corresponds to a 20-fold alanine scan as an essential tool to validate antibodies before use in R&D or clinical applications. Finally, we developed an alternative one-step solution for antibody cross-reactivity analysis based on the human epitope microarray with more than 29,000 different human epitopes as indicated in the immune epitope database. So how does a typical epitope mapping work? Well, and if antigen sequence is known, it's translated into overlapping peptides that are printed onto a microarray. The microarray with the antigen-derived peptides is incubated with a sera or antibody sample, followed by staining with fluorescent labeled secondary antibodies and readout with a microarray scanner. An epitope is highlighted by a row of peptide double spots with a consensus motif that corresponds to the epitope of an antibody, as shown on the left with the raw microarray scans for a monoclonal and polyclonal sample. In contrast to other suppliers that work with pre-synthesized peptides, we always perform epitope mapping with maximum peptide-peptide overlap for high-resolution epitope data. In a linear epitope mapping with 15 amino acids, this corresponds to a peptide-peptide overlap of 14 amino acids and a peptide-peptide shift of a single amino acid. To save peptide synthesis costs, other suppliers often reduce the overlap to 10 or 11 amino acids. However, this doesn't not only decrease the amount of data points, but also may lead to false epitope data. So as shown here for a linear epitope mapping of an autoimmune serum against autoantigen CMPA, with the maximum peptide-peptide overlap of 14 amino acids, shown on the left, we observe two nicely separated epitopes close to the N-terminus of CMPA, or SEMPA. However, with the reduced peptide-peptide overlap of only 11 amino acids, we apparently only see a single epitope, what is simply false data and we can only be found in the literature. So only with a high-resolution epitope mapping, such as PEPperPRINT, with maximum peptide-peptide overlap, you'll get reliable epitope data. From a sample point of view, we're totally open and familiar with all kinds of sample from purified antibodies, supernatant plasma or serum samples. Moreover, we can work with any sample species or antibody epitope, whatever is requested, because the technology is quite flexible. So far we talked about linear epitope mapping, but we also developed a method for conformational epitope mapping that was benchmarked with rituximab, a therapeutic anti-CD20 antibody. Rituximab binds against a conformational epitope of an extracellular 15 amino acid CD20 loop between amino acids 167 and 183 with E, S, N, and P in the binding pocket of the antibody, as shown by the crystal structure analysis, which was published in 2007 in J Biology Chemistry. So to identify the epitope of rituximab with our peptide microarray technology, we translated CD20 into overlapping linear and cyclic constrained peptides. To cover different protein conformations, we worked with 3 different peptide lengths to mimic different protein loop sizes. The incubation of the microarray with the linear CD20 peptides did not show any interaction with rituximab, as shown here with the flat line in the intensity plot. However, in the cyclic constrained peptides, we observe clear signals that increase with peptide length and hence loop size. The highest signals with the 13 amino acid peptide loops nicely reflected the natural conformation with the 15 amino acid CD20 loop. Based on the peptide microarray with the cyclic constrained CD20 peptides, we're able to identify the amino acid stretch EPANPSEK as an actual epitope of rituximab. So kind of moving on in the same case study, we wanted to take an in-depth analysis of the rituximab epitope. So we started with the CD20 wild-type peptide that contains the epitope of rituximab and printed a new microarray with a full epitope substitution scan of the wild-type peptide. As shown in the heat map, we exchanged all amino acid positions starting with the C-terminal lysine on top with all 20 amino acids. This means we did not only perform a standard alanine scan, which just corresponds the first row here in the heat map, but also a cysteine, aspartic acid, and everything. So the raw microarray scan highlighted a typical epitope, substitution pattern with conserved and variable amino acid positions. For an in-depth analysis, we quantified the microarray data and visualized the substitution pattern in a so-called amino acid plot as shown on the right. We set the intensity of the wild-type epitope to 100% and normalized all the amino acid exchanges to the wild-type peptide. This resulted in a clearly variable N-terminal stretch with a preference for acetic amino acid, what likely just resulted from the ionic effects in a highly conserved N-terminal stretch corresponding to the rituximab epitope. While amino acids N, P, and S are absolutely essential for rituximab binding, the C-terminal lysine can be exchanged by arginine without any loss in binding. Exchange of the N-terminal aspartic acid by glutamic acid was somehow tolerated, but will result in 50% less antibody binding. So this approach does not only enable the in-depth analysis of epitopes of therapeutic antibodies, but also can use the matrix information of the amino acid plot to blast for possible cross-reactions of an antibody. Moreover, if you consider that M, P, and S are essential for rituximab binding, you can also use that data for patient stratification. So if sequencing data highlights a point mutation in the CD20 position, there's no need to administer rituximab at all, since it simply won't bind. So our linear and conformational high-resolution epitope mappings combined with the subsequent epitope substitution scan gives you the clearest picture of an antibody protein interaction you can get. So in the upcoming slides, we'll discuss our cancer research application and go into some specific projects with a focus spanning basic research to neoepitope identification. So for cancer research, we provide standard peptide microarrays with selected tumor antigens or more specific melanoma antigens. We can also design a custom peptide microarray covering selected antigen peptide linear libraries as linear or cyclic peptides. And finally, our cancer neoepitope microarray, which covers personalized cancer DNA point mutations that are translated into peptide variants. And I'll explain this strategy in a little bit more detail later. So one of our first cancer biomarker studies was done with the University Hospital Heidelberg. The aim of the study was the identification of prognostic serum markers in glioblastoma patients and was published in Oncotarget in 2015. So we translated 6 known glioblastoma-associated antigens into overlapping peptides and printed them onto a first-generation peptide microarray for the primary screening. So each peptide microarray covered 2 array copies for the analysis of 2 patient sera per chip. So in the middle, this is an example of a chip the size of a regular microscope slide, and you can have 2 patients per chip, so one on the top and one on the bottom. So we initially tested 24 glioblastoma serum samples, shown on the right, from long and short-term survivors. So what we were able to identify was 30 candidate marker peptides in 4 of the 6 tumor-associated antigens. Then we moved on to printing a second-generation peptide with 16 array copies. So this is technique is quite familiar, and then we do a primary screen, and then once we can drill down into specific candidate markers, we can do a secondary generation, which saves on time, costs, and basically getting a higher resolution. So we started with two multi-center study sets with 190 glioblastoma serum samples were tested on this array. And then we used bioinformatic analysis data which result in the identification of a TNC peptide as a prognostic marker peptide for this for the long-term survivor group. So basically, we were able to correlate that a higher antibody titer against this TNC peptide correlated with longer survival of more than 36 months compared to a short-term survival of glioblastoma in 6 to 10 months. So in this study, we could demonstrate that the flexibility of the PEPperChip platforms allows for an iterative identification and validation of a prognostic tumor marker. So for another example, in a recent study published in August of this year, David Hahn from the University of Connecticut Health Center and his team described a genomic-driven immunoproteomics platform for the identification of tumor-derived antigenic targets to gain molecular insights into anti-cancer immunity. So they initially applied deep sequencing of 348 high-frequency cancer genes from 15 breast cancer patients to identify mutations in the coding regions. These mutations identified from each patient were translated into patient-specific peptide microarrays with mutant and wild-type peptides printed next to each other. On the left, you can see the peptide array of patient 1 that was incubated with serum from patient 1. The array had 460 peptide pairs in duplicate. Antibodies present in serum from patient 1 reacted with 5 mutant peptides, but not to their wild-type counterparts. Considering the high number of mutations, here it was about 460, this is only a very small number of peptides that are immunogenic. In the 15 tested patients, they found altogether 9 mutations, which invoked a strong immune response. So some mutations were shared between 15 patients. On the right, you can see 3 shared mutations of patients 1 and 6. However, only the first mutation induced an immune response in both patients. Mutation 2 and 3 only invoked an immune response in patient 6. Thus, the immune response to a mutation is highly personalized. So the reason could be that the affinity of the mutant peptides to the HLA receptors is not high enough for all mutants. So that mutant peptides are not efficiently presented on cells, or they often have to overcome immune suppression in the tumor microenvironment. But altogether, it's just illustrating that the vast amount of information you can obtain by doing this neoepitope discovery with our peptide microarrays. So for the first time, these cancer neoepitope microarrays enable the translation of sequencing data into personalized peptide arrays within 4 weeks. This novel cancer peptide microarray generation paves the way for a range of new applications in cancer research, including the development of cancer vaccines, the identification of immunogenic epitopes for cancer immunotherapy, or the discovery of prognostic cancer neoepitopes. So we're very happy to present this new data published by David Hahn and his group. And it's probably one of the company's of PEPperPRINT's highlights of this year. So in the third case study, we're addressing a link between cancer and autoantibodies. In this study, a 79-year-old woman with a 3-month history of bruising and bleeding disorders came to the hospital. A lymph node biopsy revealed mantle cell lymphoma, and a blood test factor 13 activity of less than 3%. It was suspected this essential blood clotting protein was inhibited by an autoantibody that has been produced by the lymphoma cells. So to identify the suspected autoantibody, we translated the factor 13 alpha subunit into overlapping 15 amino acid peptides with 14 amino acid peptide overlap for a high-resolution epitope mapping. The factor 13 peptide array was incubated with the patient plasma, and we identified 2 clear epitopes within the beta sandwich and the catalytic core domain of factor 13 alpha. It's speculated that binding this autoantibody to factor 13 blocks binding in the substrate and thus inhibits blood clotting. Now on to example number 4 for our tumor antigen microarray. So instead of, so this tumor antigen microarray is a custom array. It's a standard array that has 22 selected and known tumor antigens. and it's printed as overlapping peptides for a multiplexed epitope mapping. So in this case study, this array was tested with a clinical sera of a tumor patient, and we were able to identify strong IgG responses against epitopes of 2 tumor antigens, and the response against SSX2 actually exhibited a polyclonal character with multiple epitopes. As with other customer standard tumor antigen microarrays, this approach can be used to identify prognostic cancer epitopes for non-invasive or early cancer diagnosis or for patient stratification. Other standard peptide microarrays for cancer research include the melanoma antigen array, which I discussed previously, and oncovirus peptide microarray or breast cancer discovery microarray, which I'll discuss now. So this study was initiated by Duke University. We performed a highly multiplexed epitope mapping of IgG responses of 10 breast cancer patient sera with a discovery microarray with 61 selected breast cancer antigens corresponding to more than 35,000 different peptides. The goal of the study was to identify common IgG responses for the second-generation peptide microarray for a clinical trial of the viral stimulation of anti-tumor antibody responses. They wanted to push the B-cell response against breast cancer antigens or tumor cells by treatment with poliovirus. So the microarray scan on the left highlighted a representative IgG response of a breast cancer patient sera against the 61 selected breast cancer antigens, and each signal stretch in red corresponded to an individual breast cancer epitope. After data quantification, the response profiles of all sera were plotted against the microarray content as highlighted in the intensity plot on the right. In general, we observed complex IgG response profiles with excellent signal-to-noise ratio, but also with clear differences between the 10 breast cancer patient sera. So despite the varying IgG response profiles, statistical analysis of the microarray data highlighted a number of common antibody responses in 10 patient sera. The top peptide even exhibited very high statistical significance. It was found with a high antibody titer in all breast cancer sera. So for the clinical trial, we will generate a second generation peptide microarray based on the top peptide hits of the discovery screening. The corresponding breast cancer sera are presently collected. Since the study is still ongoing, the sequence of antigen data has been kept confidential. So apart from high-density peptide microarrays and synthetic peptides, we also provide custom and standard peptide pools for T-cell stimulation with varied QC levels and synthesis scale. For T-cell stimulation, we can generate 8 to 15 amino acid peptides with free amino or carboxyl termini for T-cell loading. We have access to 8 different peptide synthesizers, more than 20 RP-HPLC columns, and MALDI-TOF for peptide QC. The delivery times vary with pool size, and the synthesis scale and their requested purity, but we're happy to provide these customized peptide pools to more than 100 different customers all over the world. So the presented customer projects are a great example of how peptide microarrays can support cancer research. Certainly, they are also used in other research fields, including vaccine development, autoimmune research, infectious disease research, or peptide drug development. So we provide the most flexible microarray platform in the market, meaning that we can easily generate more or less any peptide microarray for your project. Our microarrays can contain up to 35,000 different linear or conformational peptides and are manufactured under a very strict and ISO 9001 certified quality control to ensure highest peptide quality. We provide a comprehensive support with fast response times to enable you to reach your project goals in the most straightforward and optimal manner. And in case you're not familiar with the microarray handling and analysis, we can also help you with a broad package of microarray services that include all analysis, data evaluation, and a scientific reporting according to the project needs. So right now, PEPperPRINT is working with 9 of the top 10 pharma companies, multiple biotechs, non-government organizations, and universities all over the world. So in conclusion, if you'd like more information, we have on our website a number of videos, webinars, application notes. So we highly suggest you visit us on our web or get in contact with us directly. So in summary, we look forward to learn about your project, your project needs, and see how we can work together. And I thank you for your attention, and we'll take any questions.
Great, thank you very much, Jill. So again, some members of the audience already typed some questions up. So if you do have any questions, please type it in the questions pane. So we will try and answer a few questions here. We probably won't be able to answer all the questions, but we will make sure that everyone receives an answer. So here's the first question. Can the peptides resemble disulfide bridges in the antigen structure? Maybe also our panelists here. I want to introduce our panelists, Lisa Wick.
Yes, so hello. I'm working here at the headquarters in Heidelberg, Germany, and I would like to answer the question. So yes, we can resemble disulfide structures in the antigen. So especially if the 3D structure is known, we can look into databases and then synthesize the peptides which are between, so to say, between these, or which make this disulfide bridge. We can then induce cyclization via disulfide bridges, but usually we do cyclization via a thioether formation, which is more stable than disulfides on the chip. But yes, we can do that.
Great, thanks, Lisa. So here was another question. Basically, where the peptides are coming from, whether they're encoded from a gene bank or rather where are the peptide where do we obtain the peptide sequences that we print on the chips?
I can also comment on that. So typically the peptide sequences come from the customer, meaning that when we, for example, have a project in breast cancer, usually the customer has selected antigens in mind, and then we simply translate the antigen sequence into overlapping peptides. So the antigen sequences can be found in UniProt, for example. Or there's another possibility. We can also look into databases, for example, the immune epitope database, and look for known epitopes. This is especially interesting for biomarker studies where you would like to focus more on a broad epitope coverage and not on single antigens as overlapping peptides.
Great, thank you, Lisa. Here's another question by an attendee, and he is interested in a breakdown of the cost of the slide chips. Yeah. Can anybody comment on that?
So it's really simple, the price structure here for the chips. So as Jill said in the presentation, we charge per chip and not per peptide. And for example, a chip with a microscopy slide format, which is a standard format, it can cover up to 5,500 peptides in duplicate. And the chip costs €900 or roughly $1,100. And the price for this chip is always the same. And we can either then print 5,500 peptides on the whole chip and then use one sample, or we can separate the chip area into, for example, 5 identical array copies. And so you can then do 5 analyses on one chip, and the price for the chip is still the same. It's still €900.
Great, thank you. So another question here. What is the concentration of the peptide features?
Yeah, the amount of peptides on the chip is in the picomolar range. So it's a little amount, but it's still sufficient for a very efficient epitope mapping.
Another question, what about post-translational modifications? Can the peptide be modified?
What we can do so far is we can incorporate unnatural amino acids, and we can also incorporate, for example, citrulline or homocitrulline within the peptides. What more modifications are not possible yet.
Okay, so I'm scrolling here further through the questions pane. Somebody asks a very basic questions. Do you provide pre-made peptide arrays for discovery?
Yeah, we have a list of standard peptide microarrays. You can find the list on our website under products, peptide microarrays, and they cover, for example, the whole proteome of dengue virus, or the whole proteome of influenza virus. So there should be standard arrays for infectious disease research, but we also have standard arrays for autoimmune research with known autoimmune epitopes on the chip, and but also, as Jill just mentioned, we have a standard array for tumor with tumor antigens translated in the overlapping peptide. So there's a whole list on our website.
Okay, so we'll do two more questions. Here's another one. Are your chips reusable, or are they only used once per assay?
Yeah. So in theory, they can be stripped, but of course, stripping can also influence the peptide and the quality. And so we can give out a stripping protocol, but typically we just use the chips once, because usually the customer wants to have data or results that can be compared from one to the next chip, so they want to have the best quality always. And since we can print multiple array copies on one chip, then usually you can perform 5 analyses on a chip, or even 16. This, of course, depends on the size of your peptide library. So there is a protocol, but we recommend to use a new array copy for each sample.
Great, thanks, Lisa. So last question, how do you characterize the quality of the peptides on the chip?
So what we do is we synthesize the peptides on a large glass, which covers 14 peptide microarray slides. And one of these microarray slides is always a quality control slide. And before we send out all other 14 chips to the customer, we use a reference serum and stain the quality control slide. And only if this quality control slide has the correct quality, we send out all the other chips to the customers or use them in-house for customer studies. We cannot do a direct, say, mass spectrometry or direct analysis of the peptides on the chip, because this would simply destroy the peptides on the chip. And because we synthesize the peptide directly on the spot, so we cannot do a mass analysis here on this spot. But we do it with a quality control chip, which is on, like, say, a batch control.
Yeah, great. So questions are still rolling in. I want to thank everyone for your interest. I'm going to read one more question, actually. That, I think, is an important one. Can your chips expire? How can I check if a chip is still viable after not being used for some time?
Yeah, so the chip quality is very stable. You can use them over years. We also use the chips 1 or 2 years later, because it's a dry material. We add inert gas on the in the box where the chips are in, and we found that they are really stable. So it means that, for example, if you have a chip with 5 array copies, you can stain the first array copy on day 1, and then the next copies 3 or 5 months later, it doesn't change the quality of the arrays or the peptide.
Great. Well, thank you all for your interest. So I think I'm going to stop our discussion here. So for anybody who has not received an answer, we will answer you offline directly. So keep on typing those questions. So I want to thank everyone for attending this webinar presentation. We also want to thank our speaker, Jill Rowan. We will make the presentation slides and the webinar file available shortly. If you do have any further questions, feel free to contact the PEPperPRINT team directly. And please check out our website for updates, future events, and webinar announcements. I will end this webinar now. Thanks again, everyone, for dialing in.